like I said, there's quite a few slides to get through. We are talking about the chemistry of life. We can't talk about life without talking about the chemicals that make us up first. So part one is carbon. Carbon is what makes something organic. We are made up of organic compounds. All living things are. So a cell is mostly water. The rest consists mainly of carbon-based molecules. Carbon forms large, complex, and diverse molecules necessary for life's functions. Organic compounds are carbon-based molecules. And note the use of organic in science versus the use of organic in the grocery store. When you go to the grocery store, you go to the organic section, it means that something was grown without pesticides. That's what organic means in the grocery store. But in science, it just means a carbon-based molecule or a carbon-based organism. We are a carbon-based organism, so we are considered organic, whether we use pesticides on ourselves or not. Carbon is a versatile atom. It has 4 electrons in an outer shell that holds 8 electrons. And carbon can share its electrons with other atoms to form up to 4 covalent bonds. You did your Edpuzzle last week on the different bonds, ionic, covalent, hydrogen bonds. This week we are focusing on carbon and the bonds that it can make. So it can make 4 single bonds. It can make 2 single bonds and 1 double bonds. It can make 1 single bond and 1 triple bond with something. Carbon can use its bond to attach to other carbons and form an endless diversity of carbon skeletons varying in size and branching patterns. On the top, you have 1 branching pattern where there's a bunch of carbons all put together. And then there's hydrogens branched off, and one end you have a C double bonded to an O. And then on the other side, it's bonded to an OH. That's called a carboxyl group. It's called a functional group. You guys are going to be doing different functional groups in your lab this week. B, on the bottom, you can see there's your carboxyl group. And then on this side, with an N, that's called an amine group. So it makes of amino acids. So there's your carboxyl group there. So this is the kind of stuff you're going to be doing in lab this week. You're going to be looking at molecules and you're going to be picking out the different functional groups. You do have background reading on it. And then the OHs here are called hydroxyl groups. So that is in your lab. It will be in your lab readings, and you'll be picking these out this week. Stop annotating. All right, the simplest organic compounds are hydrocarbons, which contain only carbon and hydrogen atoms. The simplest hydrocarbon is methane, a single carbon atom bonded to 4 hydrogen atoms. It is also one of the molecules that is causing our climate so much trouble, is methane being put in the air faster than we can process it. And methane looks, these are 3 different ways to illustrate a methane molecule. On the left is called the structural formula. If you're just writing it on a piece of paper, that's usually how it looks. You have the carbon bonded to 4 hydrogens. The ball and stick model is what we would be making if we were actually in class. We have little balls and sticks. The sticks represent the bonds. Different color balls represent the atoms. And then the space filling model, we don't do that very much, but you'll see the first 2 in pictures. Each type of organic molecule has a unique 3-dimensional shape. The function of organic molecules relate to their structure. The unique properties of an organic compound depend on its carbon skeleton. Can be either straight or ringed, like we saw the big, long, straight, or it can be in a ring. And then the atoms attach to that skeleton. The groups of atoms that usually participate in chemical reactions are called functional groups. Two common examples are hydroxyl groups and carboxyl groups. That's what we looked at a couple slides ago. And many biological molecules have two or more functional groups. And there's a crash course on carbon if you'd like to watch it. That is not what our crash course is about this week. So part 2, macromolecules. Macro means large as opposed to micro meaning small. And then molecules. So we're talking about large molecules that a lot of them we eat, but we eat them so we can make up structures in our bodies. But we are made of all of them. So giant molecules from smaller building blocks. On a molecular scale, many of life's molecules are gigantic, earning the name macromolecules. Most macromolecules are called polymers. Polymers is a molecule that's made by stringing together many smaller molecules called monomers. So it looks like a chain, like on the bottom. Mono means one. We've learned that in the past at some point. So you put 1 plus 1 plus 1 plus 1 plus 1 together, you get many. Poly equals many. So a polymer is just a certain molecule that's made of many monomers. It's illustrated at the bottom. One of those little hexagons is called a monomer. You put a bunch of them together. The big long chain is called the polymer. Organisms have to break down macromolecules. The process of digestion breaks down macromolecules to make monomers that are available to your cells. So we eat protein, which is a huge monomer, or carbohydrates, which is a huge monomer. And then it goes through our digestive system and it breaks down into the individual subunits because our cells can't use them until they are broken down. And then the breaking of bonds. When food is digested, the bonds holding the atoms together in food molecules are broken, and this is what gives us energy. So the longer the molecule is that we eat, the more energy it's storing for our bodies to use. Dehydration synthesis. This is a process that removes water in order to combine molecules, or to combine monomers. So dehydration, everybody knows what it means to be dehydrated. If you're dehydrated, it means you're lacking water. And then synthesis means to combine. So it's a process that breaks bonds between monomers. By removing a molecule of water, it allows those two molecules, A and B, to bond. The opposite is called hydrolysis. Hydro means water. You're using water to lyse or split a molecule. So by inserting a molecule of water, it splits up those compounds. These GIFs only run once. If you refresh your page, because you guys all have these, you can see how it works in an animation. There's 4 categories of large biological molecules. 3 of them, that, like I said, that we eat to build structures in our bodies: carbohydrates, proteins, lipids, we all eat those, and then nucleic acids. That's our DNA that our cells have as genetic material. We'll talk about that a little bit, but there's a whole chapter dedicated to DNA later. So we're just introducing it today. carbohydrates. The name literally means carbon plus water. Carbo plus hydrate, water. These include monomers and polymers of sugar. Most names of carbohydrates end in OSE. I know when we think of carbohydrates, we think of pasta and rice and tortillas and bread. But as far as our bodies are concerned, your body processes them like they would process a sugar. So they include small sugar molecules like sucrose. Sucrose is table sugar that you all probably have in your house and use to sweeten. I mean, I use it to sweeten my coffee, but you can use it to sweeten anything. But it also includes long starch molecules like in spaghetti, French fries, bread. So the monomers are the small ones, the individual units, glucose, fructose, then you start putting them together. You put glucose and fructose together, you get sucrose. That's what our table sugar is made of. But then you see the starch on the bottom is made of huge long chains of glucose molecules over and over and over again. So there's simple sugars and complex carbohydrates. In animals, carbohydrates are a primary source of dietary energy and raw material for manufacturing other kinds of organic compounds. In plants, carbohydrates serve as building material for much of the plant body. In plants, their sugar is called cellulose, still ends in OSE. Monosaccharides are the monomer of carbohydrates. Mono means one, saccharide means sugar. And there's that CH again that sounds like a K. Always science words, when you see CH, it sounds like a K. So monosaccharides are simple sugars that cannot be broken down by hydrolysis into smaller sugars. They are also the monomers of carbohydrates, and you have three main monosaccharides: glucose, fructose, and galactose. And I know they all kind of look the same, but if you look at their branches, fructose has a branch right here. And then galactose, let's see, where's the difference? Oh, for galactose, these two, and then these two are different. So they're very, very, very slight differences in the way the atoms are arranged in these three monosaccharides. They have the carbon skeleton, this is what we talked about, but this is a ring-like carbon skeleton, but their branches are just a little bit different. Disaccharides. Di equals two, and then saccharide again means sugar. These are constructed from two monosaccharides and formed by a dehydration synthesis reaction. Water gets pulled out in order to synthesize or combine the two monosaccharides. So if you put two glucose molecules together, you get maltose. If you put a glucose and a fructose together, that's when you get sucrose or table sugar. If you put a glucose and a galactose together, you get lactose, which is the sugar you find in milk. So there's lactose in milk, maltose in beer, malted milkshakes, and malted milk ball candy like Whoppers. If you've ever had Whoppers before, those are malted milk ball candies. And then sucrose is your table sugar. And then polysaccharides. Poly means many, saccharide, we've gone over it twice, means sugar. Also known as complex carbohydrates. Made of long chains of sugar units. They're the polymers of many monosaccharides bonded together. Starch is a familiar example of a polysaccharide. It's used by plant cells to store energy. It consists of long strings of glucose monomers. So that is a microscopic image of a starch granule in potato cells. If we were in class, we would be staining them purple and looking at them under a microscope. But if you look at its chemical compound, it is just repeating units of glucose molecules, strung together in a huge long chain. Glycogen is a polysaccharide used by animal cells to store energy. It gets converted to glucose when it's needed. And out of the two reactions, which reaction will split this molecule into smaller ones? We went over dehydration synthesis and hydrolysis. Which one splits the molecules? You guys can answer me or you can think about it, but it'll be a question later for you. We have a lot of glycogen in our liver. It's where our glycogen is stored. When we need energy, our body takes that glycogen out and splits it up into glucose so we can use it. Cellulose forms cable-like fibrils in the walls that enclose plant cells to form wood and other plant structures. Its glucose molecules are bonded in such a way that it cannot be broken apart by most animals. So while we eat parts of plants, we don't eat like bark and branches in the wood because our body can't break that down. We stick to the leaves and the plants and the flowers and the fruits. In our diet, this is called fiber. It keeps our digestive tract healthy. So what little we get, everybody should eat more fiber. Polysaccharides, almost all carbohydrates are hydrophilic or water-loving. Monosaccharides and disaccharides dissolve readily in water and form sugary solutions, which we all know because we all probably drink sugary drinks. Cellulose does not dissolve in water, but water can still stick to its surface. Lipids are the second class of macromolecules that we eat. They are hydrophobic or water-fearing. If you have a phobia or something, you are afraid of it. So it does not mix well with water. These are neither macromolecules or polymers because they are not made of a chain of the exact same monomers repeated over and over, like carbohydrates. So they are needed in our diets, and they are large molecules, but they're not considered polymers because they're not made of the same identical repeating units over and over and over again. Fat is one type of lipid. A typical fat is called triglyceride. If you break up the word triglyceride, you can figure out what it's made of. A glycerol molecule joined with 3 fatty acids. That tri, just like mono means 1, di means 2, tri means 3. You're riding a tricycle, it has 3 wheels. And then there's another reaction question. Which one of our reactions, dehydration synthesis or hydrolysis, would join or combine all of these molecules together? You can take a guess or make sure you know it for your lab and your test later. Functions of fat. Our bodies need a certain amount of fat to perform essential body functions. Fat gets a bad rap. Yes, you're not supposed to have too much of it, but your body does need some fat to be able to function properly. Energy storage. So how does our body get energy? It eats. And if you look at the molecule, you can see how much energy is stored by how long it is and how many bonds it has. Fat stores our body or cushions our organs against trauma. So if we fall, we don't automatically bruise our kidneys. There's a little cushion of fat in there. So if you don't fall too hard, you don't actually get hurt. It also insulates us to help maintain our body temperature. Body temperature is huge. Especially right now with the coronavirus, everybody wants to stay at that temperature or else you're at the risk of being sick. Structure of different kinds of fats. If the carbon skeleton of a fatty acid has the maximum number of hydrogens, it is called a saturated fat and its structure is straight. If the carbon skeleton of a fatty acid does not have the maximum number of hydrogen, it is called an unsaturated fat and its structure is bent because it will have a double bond in the middle. You can see that illustrated in the picture. Saturated fatty acid, you have that carbon stripe in the middle, all the C's. They're all single bonded to each other. It's called a saturated fatty acid. Unsaturated is when you have a double bond and it looks bent. Animal fats have a high proportion of saturated fatty acids. They can easily stack, tending to be solid at room temperature, like butter and bacon fat. They contribute to atherosclerosis, in which lipid-containing plaques build up along the inside walls of blood vessels. You can think of butter being in its solid state, just stacking up inside your blood vessels. So saturated fatty acids are the bad ones because you don't want that. Plant fats are high in unsaturated fatty acids, and they tend to be liquid at room temperature. Since they have that bent shape, they can't stack to become solid. These are the good fats: avocado, olive oil, fish oil. These will stay liquid in your bloodstream. A second type of lipid is steroids. They're very different from fats in structure and function. The carbon skeleton is bent to form 4 fused rings. So you can see in the picture, you see 4 rings connected together. Doesn't matter the technical shape, just whether it's a pentagon or a hexagon, just that they're ring shaped and they're all stuck together. And they just vary in the functional groups attached to the set of rings. And these chemical variations affect their function. So they're going to have branches. They're going to have branches coming off here with different molecules. And whatever the branches are is going to tell you what the function is. Cholesterol is a key component of cell membranes. Cholesterol is something else that gets a bad rap, just like lipids in general. The base steroid from which your body produces other steroids, such as estrogen and testosterone. So cholesterol is necessary for your cell membranes to keep them together and functioning properly. And as hormones, we all need hormones. We need a certain amount of hormones throughout our bodies at different times in our lives. Proteins is the third class of macromolecules. You can see they're made of individual amino acids, which we'll get to. Proteins are polymers constructed from amino acid monomers. They account for more than 50% of the dry weight of most cells. They perform most of the tasks required for life and they form enzymes, which are chemicals that change the rate of a chemical reaction without being changed itself in the process. We have a whole chapter or section on enzymes in one of our upcoming chapters. Major types of proteins, you have structural proteins, storage proteins, contractile proteins are actual muscles to help move us, transport proteins, and then your enzymes. So there are a lot of different classes of proteins that are vital for life. Amino acids, all proteins are macromolecules constructed from a common set of 20 kinds of amino acids. And this part is going to be important for your lab this week. Each amino acid consists of a central carbon, which you can see, it's pointing to it, bonded to 4 covalent partners. 3 of those partners are common to all amino acids. So all amino acids have a carboxyl group, that's the pink, an amino group, that's the green, with the N, with the nitrogen in it, and then a hydrogen atom on top. Every single amino acid is going to look the same for that portion. The 4th partner is different in all 20 amino acids. It's either called the R group or the variable group. And that R group, that side chain, will determine the name and the function and the properties of that amino acid. The example is the box. Those chains make it a positively charged R group, which is going to interact differently with something negative or something acidic or something basic. And those are just 3 of the 20 examples. A polypeptide is a chain of amino acids held together by peptide bonds. A functional protein is one or more polypeptide chains that are twisted, folded, and coiled into a uniquely shaped molecule. Proteins consisting of 1 polypeptide have 3 levels of structure: primary, secondary, and tertiary, first, second, and third. It's pretty much the same in English and in Spanish. And proteins consisting of more than 1 polypeptide chain have a 4th level called the Quaternary Structure. Primary structure, your body has thousands of different kinds of proteins. They're all made of the same 20 amino acids, just like the alphabet with only 26 letters. How do we get a million different words? You just rearrange the letters in different ways, in different orders, different lengths. Some words are 2 letters long, some words are 13 letters long. Some proteins consist of 3 amino acids, some proteins consist of 50 amino acids, and they're all in different orders. So proteins differ in their arrangement, their sequence, their order, and length of amino acids. And the specific sequence of amino acids in a protein is called its primary structure. It would just be like the way the letters are arranged in a certain word is called its primary structure. This is just an example of amino acids. There's 129 amino acids in this protein. And you can see the amino acids. They're abbreviated, obviously, for obvious reasons. And the order of them determines the specific protein. A slight change in the primary structure of a protein affects its ability to function. The substitution of 1 amino acid for another in hemoglobin causes sickle cell disease, which is an inherited blood disorder. We'll talk about sickle cell disease a lot. So in normal hemoglobin, that middle protein, that middle amino acid is supposed to be glutamine. But if it gets mutated in just the right place, it turns into a valine and screws up the whole structure. Secondary structure. The primary amino acid sequence will fold or twist in such a way to make either an alpha helix or a beta pleated sheet. So you have the primary structure, just the order of amino acids. The way they interact with each other will form one of these two shapes. And this is called the secondary structure. The tertiary structure is the overall 3D shape of the polypeptide molecule. It's reinforced by interactions between R groups of different amino acids with the polypeptide. You can see in the picture, you can have hydrophobic interactions where they're repelling each other, disulfide bonds, a salt bridge, hydrogen bonds. There's a lot of different interactions that can happen in the tertiary structure that will determine how the protein interacts with itself, whether it folds or it's kind of a straight line or loops around. And the quaternary structure forms when more than two polypeptide chains are present and bonded in a certain way. There's kind of the overall primary, secondary, tertiary, and then quaternary interacting with each other. Protein shape is very important. A protein's three-dimensional shape typically recognizes and binds to other molecules, and it enables a protein to carry out its specific functions within a cell. If a protein loses its shape, it's called denaturation, and it doesn't work anymore. And that is bad because we need proteins to pretty much do everything for us inside of our bodies. A protein shape is sensitive to the surrounding environment. It can be affected by temperature, by pH, and by ionic or salt conditions. A negative change in any of these results in denaturation, like I just said, which it unravels and loses its shape and therefore also loses its function. High fevers above 104 degrees in humans can cause some proteins to denature and lose its shape and its function. So part of the reason our bodies maintain a steady temperature throughout our lives is so our proteins function properly. So it can perform all its little tasks in our bodies. Misfolded proteins are associated with not only sickle cell disease, but Alzheimer's disease, mad cow disease, and Parkinson's disease. And the nucleic acids is our last one. This one will be short. Like I said, we have a whole chapter dedicated to nucleic acids later. So for now, nucleic acids are polymers that are made of monomers called nucleotides. They store genetic information. They provide the directions for building proteins. We know how important proteins are now. And include DNA and RNA. We have DNA in our cells. The coronavirus is made up of RNA. The vaccine is made up of a piece of RNA from the coronavirus that will make a part of it and help our bodies recognize it and build up antibodies against it. DNA resides in long fibers called chromosomes. A gene is a specific stretch of DNA that programs the amino acid sequence of a polypeptide. And the chemical code of DNA must be translated from nucleic acid language to protein language to be able to make proteins to perform all those functions in our bodies. And there's the crash course on the biological molecules, which is what your Ed puzzle's on this week. So don't watch that yet. Watch it when you do the Ed puzzle.